In this video, I want to introduce you to my PhD project within the FIDELIO Consortium, an EU-funded international training network. The network consists of 14 PhD students who want to describe and unravel the mystery of bone disease and diabetes. My project is conducted at the University of Southern Denmark in the laboratory of KMEB and is entitled Bioenergetics of Bone Cells. We hypothesize that type 1 diabetes impairs mitochondrial function, and that patients with type 1 diabetes accumulate mitochondrial defects during disease progression. Furthermore, we think that mitochondrial function is key for bone health, and functioning mitochondria are key for strong bone, and mitochondrial dysfunction weakens bone and increases fragility fracture risk. Combined, these two hypotheses lead to the idea that type 1 diabetic patients accumulate mitochondrial defects, which may in part explain the weaker bone phenotype in type 1 diabetes. To test our hypothesis, we used cultured cells and cells derived from type 1 diabetic patients. You want to see how these cells look like? Take a glimpse into the microscope. We use technologies such as extracellular flux assays in the seahorse analyzer to quantify mitochondrial respiration. Furthermore, we want to test mechanistically how important mitochondrial function is for bone cell development and bone cells using a tool to increase mitochondrial damage in cellular and animal models. We use samples from type 1 diabetic patients to describe the bone microarchitecture, genetics, and biomarkers, as well as the mitochondrial function of the bone cells. If our hypothesis turns out to be true that type 1 diabetic patients increase mitochondrial dysfunction during disease progression, this could lead to a clinic implementation to supplement and treat type 1 diabetic patients with supplements enhancing mitochondrial function, leading to stronger bone and decreasing fragility fracture risk. If you are interested in that topic, follow us on social media on Twitter, Facebook, and Instagram. I want to thank the Novo Nordisk Fund for financial support and our collaboration partners at the University of Geneva, ETH Zurich, and Tamirna in Vienna.